Welcome to this explainer. Today, we're diving into a fascinating set of handwritten notes to explore a universe that is literally right under our noses, yet entirely beyond human sight. We're talking about the nanoscopic architecture that dictates how all life on Earth actually functions. So, let's just jump right in, shall we?
Okay, let's kick things off with the most fundamental question about our existence. Think about it. When you break down a towering redwood tree, a massive blue whale, or hey, even yourself, into the absolute smallest living puzzle pieces, what do you get? You hit the exact same foundational starting point every single time. And that starting point is the cell. It's the absolute baseline unit of every single living organism on the planet. But, just knowing that fact is one thing. Understanding how humanity actually uncovered this invisible biological architecture, well, that's a whole different story.
Take a look at this timeline, because it brilliantly captures how this all came together. Back in 1838, a botanist named Schleiden made this massive breakthrough, realizing that all plants are built from cells. Then, literally just 1 year later in 1839, Schwann comes along and connects the dots for the rest of the biological kingdom, proving animals are constructed the exact same way. It was this incredible 1-2 punch that completely rewired our understanding of life.
But to actually make those discoveries, that required a massive technological leap. I mean, you can't study what you can't even see, right? Early biologists had to invent completely new ways of looking at the world to finally pull back the curtain on this hidden structural universe. And these meticulous notes guide us right through how they did it.
Basically, we have two main tools in our arsenal. We started out with light microscopes, which, as the name suggests, use standard light as the radiation source to illuminate the unseen. But the real game changer? That was the electron microscope. By swapping out light for a beam of electrons, scientists could suddenly zoom in exponentially deeper, revealing the architecture of life in just unprecedented detail. But here's the catch. Just looking through a powerful lens isn't actually enough. Why? Because a lot of cells are completely colorless and totally transparent. So to actually see them, biologists have to prep the specimen. They stain it, carefully sandwich it under a super thin sheet of glass called a cover slip, and only then can they capture what's called a photomicrograph, which is essentially just a photograph taken through a light microscope.
And the whole secret sauce here is magnification, which, simply put, is just the number of times an image of an object is blown up compared to its real physical size. It's how we take the invisible and make it visible.
Now prepare yourself for a little bit of scale shock, because the numbers we're dealing with here are wild. We're moving away from millimeters, which we can easily see, down to micrometers. That's 10 to the negative 6 meters. And then we're plunging all the way into the unimaginably small nanometer scale, sitting way down at 10 to the negative 9th.
To really put that math into perspective, consider this. An entire bacterial cell is only about 1 single micrometer across. Think about that for a second. That is a fully functioning living organism existing in an incomprehensibly tight space.
And it gets even more mind-bending. Inside a cell, you have structures like the ribosome sitting at a diameter of just 25 nanometers. It's wild. It proves that inside the already tiny microscopic world, there's an entirely separate, even smaller nanoscopic universe operating at the exact same time. So, at this incredibly tiny scale, life basically splits into two distinct architectural camps. On one side, you've got prokaryotes, like bacteria, which are simpler and operate without a membrane-bound nucleus. On the other side, you've got eukaryotes. This includes animals, plants, and fungi, and they upgrade their security with a highly protected membrane-bound nucleus. To really see how these more complex eukaryotes are put together, let's take a look at the outer fortifications of a plant cell, exactly as they're detailed in these notes. So, a plant cell is surrounded by this highly resilient structure called the cell wall, located just outside the cell's surface membrane. It's built from a tough polysaccharide called cellulose, and sometimes it's even reinforced by lignin to make it stronger. What's cool is that while it gives the cell a very rigid, definite shape, it's also freely permeable, meaning certain molecules can just pass right through. But why does a plant even need this tough outer wall? Well, it's not just there for looks. It is the ultimate structural defense mechanism. You see, when a plant cell takes in too much water through a process called osmosis, an immense amount of internal pressure builds up. This rigid wall literally stops the cell from popping like a water balloon under that extreme stress, absolutely crucial for survival.
That being said, it's not a totally closed-off fortress. Plant cells actually need to communicate and trade materials with each other. They do this through these fascinating structures called plasmodesmata. These are fine strands of cytoplasm that form pore-like tubes connecting neighboring cells together to create highly controlled microscopic supply chains.
Now, moving just inside that sturdy outer wall, we hit the cell membrane. If the cell wall is the brute structural support, the cell membrane is the intelligent security system. It acts as a partially permeable barrier. This means it actively controls the movement of materials back and forth, deciding exactly what gets VIP access inside the cell and what has to stay out. Once you pass through that security barrier, you're officially in the living essence of the cell. Biologists have specific names for this stuff. The term protoplasm refers to literally all the living material inside the cell. But if you're talking about all the living material specifically outside the nucleus, well, that's called the cytoplasm.
And floating all throughout this cytoplasm, you'll find organelles. These are structurally and functionally distinct parts of the cell, and they're separated from the rest of the cytoplasm by one or two of their own membranes. When biologists map all these different common structures out together, they call it a generalized cell. It's like a blueprint of a microscopic city.
A perfect example of one of these hardworking organelles is the vacuole. You'll find these massive storage tanks mostly in eukaryotic plant cells. They're bounded by a single membrane called a tunnelplast, which acts like a gatekeeper controlling osmotic exchange between the vacuole and the cytoplasm. Inside, it stores a treasure trove of vital biochemicals, everything from pigments and enzymes to essential gases like oxygen and carbon dioxide, plus mineral salts and sugars.
All right, we are finally arriving at the very heart of this microscopic city, which is beautifully detailed right at the bottom of these source notes. We're talking about the ultimate command center that orchestrates this entire complex operation.
Enter the nucleus. Present in complex eukaryotic cells, this organelle is essentially a highly protected vault. It's surrounded by not one, but two membranes that form the nuclear envelope. And inside this vault lies the cell's master instructions, DNA, or deoxyribonucleic acid. This is the blueprint that controls absolutely every single activity happening within the cell. Peeling back the final layer inside this vault, we find chromatin. This is a dense, intricate mix of DNA, proteins, and a little bit of RNA. Most of the time, it just kind of hangs out, but right when the cell is preparing to divide, this chromatin coils up super tightly into distinct structures called chromosomes. And get this, they actually only become visible during that brief, incredible window of cell division.
Wrapping up this explainer, we've taken a truly staggering journey today. We started at the sturdy cellulose walls of a plant cell, pushed through the security barriers, and traveled all the way down to nanometer-scale DNA and those tiny 25-nanometer ribosomes orchestrating life from the shadows. Which really leaves us with one provocative thought. If this much complex, highly organized architecture is happening at a scale we literally can't even see, what other invisible secrets are hiding in the microscopic universe, just waiting to be discovered?